Test 1, 2, 3, 4, 5. Test 1, 2, 3, 4, 5.
Welcome to tonight's segment of the Adams State University Faculty Lecture Series. For those of you who do not know who I am Jessica Lardy. I am professor and director, and I coordinate the lecture series here. So thanks for attending tonight's lecture. Keep an eye out for upcoming lectures. You can even grab a RAC card over there on the way out, if you so choose. Next lecture is next Thursday, November 21st, featuring Dr. Colleen Schaffner of Psychology. So tonight's event is presented by Dr. Mai Awad, Assistant Professor of Biology from the School of Science, Technology, Engineering, and Mathematics. Dr. Awad is an Assistant Professor of Biology in the STEM School. She has previously worked at Colorado State University in the Department of Environmental and Radiological Health Sciences, and as a project assistant for several projects focused on women in the sciences and inquiry-based science education. With a background in ecology, Dr. Awad has co-authored 5 publications. She received her bachelor's degree from Jordan University of Science and Technology, a Master of Science in Environmental Science and Management from the University of Jordan, and a PhD in Ecology from Colorado State University. Tonight's lecture is titled Synergistic Toxicity of Pesticides and Heavy Metals in Apis mellifera L and Implication for Human Health. And in her words, the global decline of Apis mellifera L populations over the past 2 decades has raised concerns about the impact on pollination services and ecosystem health. The study examines the role of chemical stressors, particularly pesticides and heavy metals, on bee populations and their potential effects on human health. Findings from her study underscore the complex interactions between environmental pollutants and biological systems, highlighting the need for a One Health approach to address the interconnectedness of environmental, animal, and human health. Further research is required to elucidate the toxicological mechanisms of these combinations, including at concentrations below regulatory limits, to inform risk assessments and policy decisions. So help me in welcoming Dr. Awad.
Hi, and thank you for putting the effort to come here, although it's cold outside. Yeah, so my name is Mai. This is my topic on the synergistic toxicity of pesticide and heavy metals, and Apis mellifera, which is honeybee, and the implication for human health. It's not working. Well, you have the magic touch. I'm not. So, a bit of outline of what I'm going to talk about today. It's giving a little brief background on the research, and the first section of the research, or part of the research, will focus on the spatial variation of pesticide and heavy metals in bees products, pollen and honey, and the other section on the combinatorial impact of these toxics and on human cells. So, bit of background on the research. The honeybees population are declining. We keep yapping about that, but it's not only honeybees, but other insects, as you can see here, and other vertebrates are declining as well. However, our concern is mostly about honeybees because they've been always considered as biological indicators on the environmental stressors or environmental pollution in general. So, what is interesting about the declining of honeybees is a recent phenomena that has been discovered in the beehives called the colony collapse disorder, which means that you go to the hive and suddenly found nothing in there except just few nursing bees and few larvae, lots of food, but the queen is gone and other bees are gone as well. So almost the hive is empty. That's why we call it colony collapse disorder. So there are many factors that have been associated with the declining of bees. Pesticides as a chemical stressor. So there are many other stressors, could be biological or physical. Biological, like different pathogens, different viruses, might also enhance the declining of bees, as well as climate change and habitat fragmentation that can enhance the declining of bees. But we are focusing here in the research on chemical stressors like pesticide and heavy metals. So first, with the pesticide, since the banning of DDT, we start using other form of pesticides called neonics or neonicotinoids, which are type of insecticide that target the nervous system of the insects. So these type of insecticides in this research have been found in a global collection of honey samples from all over the world, which is really alarming. And fun fact, this group of insecticides have been banned in Europe recently because of being found and the impact that have been studies on this group of pesticide on non-direct animals and non-direct insects like honeybees. So, okay. So for heavy metal, it's another group of chemical that can enhance the that can cause the death or the continuous death of honeybee population, but heavy metals have not been studied widely as pesticide. It's not been the focus of group of chemicals. So, but this research investigated the impact of heavy metals in general on the honeybee population, and they found that it's impacting their cognition, behavior, and foraging behavior. This way, the honeybees will not find their home back because of heavy metal. So we take this into consideration as well. So what we do in this research is collect honey samples and pollen samples from different beehives. We establish we consider beehives that located in agricultural area and in urban area to see if there was a difference in these chemical in these sites. So we collected pollen and honey. Taking into consideration that bee can fly up to 3 kilometers, I think it's more than 6 miles. So we consider carefully the volunteered beekeepers and their hive, because we didn't want to have a hive that is located next to an urban area. So this is how the agricultural site look like, and this is how an urban area look like, based mainly on population number and agricultural activities. So started collecting pollen samples, getting stung many times, to the degree that when I get back home and found one ear is bigger than another because of a honeybee stung me. So we connected a pollen trap in the entrance of the beehive in order to collect pollen. And by the end of the season, we collected honey samples and freeze it until the time of analysis. As a result, we collected 61 pollen samples and 21 honey samples by the end of the season, like in September and October. And we found 38 chemicals that's classified as pesticide, whether it was insecticide, acaricide, herbicide, or a pesticide synergist. We found less pesticide in honey samples, and that can be due to different reasons, like in honey, it goes under fermentation, so the pesticide can break down. So that's why we see less chemical in honeys comparing to pollen. For heavy metals and the trace metal selenium, it's not a heavy metal, it's a trace metal. So we found arsenic, cadmium, and lead in most of the sample collected, but the selenium, to the surprise, it was more in urban area versus agricultural area. However, the overall, like this, there is no significant variation. However, the selenium is higher than other heavy metals have been found. And this was found, the analysis were performed over pollen samples. Fun fact, Colorado soil is rich in selenium. That's why you can find it in these samples. We couldn't find any significant variation in pesticide between urban and agricultural hives. However, we found them, so they are there. Okay, so to find out if the concentration we found in pollen and honey samples inducing an impact or having an effect on honeybee population, we were merciful to our bees, so we didn't test them directly, but we compare our concentration to existing literature that directly use these chemicals that we found on bees. So if you take a look at the numbers here, what we found in pollen and honey for different type of pesticide, including the imidacloprid, for example, which belongs to the neonic, it's not posing that kind of lethal risk or the risk that can impact the foraging behavior of bees. It's way less than the actual concentration that's posing a harmful impact on bees. However, if you can see here for arsenic and lead, what we found in here in pollen or in honey, the maximum concentration that we found in comparable with existing study, so this concentration is higher here than this concentration that can start causing the slowing down and learning and impacting the foraging behavior. So this is alarming. Pesticide was not that much, but heavy metals is really significant in terms of impacting honeybee population. So how we connect what we found into human health. So we took the most presence pesticide and one pesticide synergist, and this pesticide synergist was found in all samples. It's not a pesticide on its own, but it's used to activate the work of pesticide. So we take imidacloprid, which belong to the neonic, and we take our selenium and lead, which were found in high concentration, and along with piperonyl butoxide, the pesticide synergist, and we apply it to human cells to find out how they will interact together, because all of the research been focusing on a chemical on its own, but not how they interact together. And this chemical is not found alone in environment. It exists with other things. So we performed different analyses on CHO cells, and CHO is Chinese hamster ovary cells. at the beginning, I mentioned human cell. So honestly, I was working on a human cell, the cancer cells, breast cancer cells and breast normal cells. But I found out that it took me so long time to grow these cells. So instead of that, I used Chinese hamster ovary cell. It's a mammalian cell as well, and it can tell you how these chemicals interacting in a human cell. So there is no need to use actual human cell. So these type of cell, the cell analyses that I used, the clonogenic and cell survivor, the oxidative stress, sister chromatid exchange, apoptosis, and immunofluorescent microscopy and quantification of gamma H2X foci. So I tested peproneal butoxide, this is the PPO, on 3 different kind of cells, the CHO, the Chinese hamster ovary cell, and 2 other variants of the Chinese hamster ovary cells called the BL10 and the PADR9. I used these type of cell as well to find out what is the toxicity pathway. If it's on oxidative stress, then PL10 will decline. If the toxicity pathway was on genetic breakup, it will be the PADR9. So each one of those is sensitive, have a sensitive pathway to die. That's why I use different variants. So what you can see here with PADR9 was dying the most because of peproneal butoxide, which means this can be a genotoxic. And fun fact, this guy, the PPO, was reported by the EPA a long time ago as non-genotoxic chemical. So I use it as one here alone, and I use PPO along with selenium. We've been always told that selenium is good for us, right? And it's good for bee, but at certain concentration, it can be toxic. So selenium with PPO enhance the toxicity in PADR9 cell. Here you can see the colony formation. It's when the cell grow together to form colony. This is the control. This is under the application of lead. And this is with peproneal butoxide. You can see in PADR9 there is no colony almost. And this is a combination of peproneal butoxide with lead. You can see there are none of these colonies, which means it's synergistic. It has a synergistic toxicity. They are dying more in here comparing to this one. Okay. Apoptotic means the programmed cell death. It's simply whenever the cell is having problem with regenerating and regrowing. So they have a self-instruction for themselves, just die. You can't fix yourself. So this is apoptosis. So here we found that they are dying because of peproneal butoxide, and there is a significant impact. And here with other combination, peproneal butoxide and these chemical alone, as well as when you combine them with other chemicals like selenium, lead, and imidacloprid, you have a significant impact, higher significance or higher apoptotic rate in the variant of PADR9. And this is these are picture of where the arrows are pointing to apoptotic cell. It's the abnormal shape of cell. And you can see under high concentration of PPO how they are dying. using another test to find out if this chemical is genotoxic. There is no significant impact, but we still found there is a break in DNA under the sister chromatid exchange assay, but not as we were hoping for. And another way to find out if this chemical is genotoxic is by counting the fraction of gamma H2X, which is a phosphorylated protein of H2X when the DNA is under or being exposed to a damaging material. This H2X will be phosphorylated to the gamma H2X, and it will start to giving signal that the DNA is under damage. So counting this, having more of gamma H2X means you have more damage in the DNA, and you can see how it was significant to see the impact of piperonyl butoxide on the damage of DNA in both CHO cells and BADR9. And you can see here the combination is true. Was not significant to the piperonyl butoxide by itself. However, you can still see the variation of these chemicals as alone and as a combination in comparing with the control. So it's still happening. And here is a picture of the gamma H2X under different concentration of, it's one concentration actually, of PPO on its own, the piperonyl butoxide, and how it's completely damaged here. And the other variable, the BADR9 cells. So these lights that you can see, it's an indicator of DNA damage caused by this chemical. So in conclusion, we found these chemicals, the pesticide and heavy metals, and some of the honey and pollen samples. Pesticides were under the limit of, or under the safe, what we call a safe, but there is a probability of having these pesticides interacting with other pesticides or with other heavy metals, which can have a synergistic impact and imposing risk for both human and honeybees. There were no spatial variation to the surprise between urban and agriculture. We always blame agriculture of having more pesticide, but we found it with no significant variation. We need more research to investigate the safe limit of pesticide along as with heavy metals, and we found that PPO was toxic, not just cytotoxic, it was a genotoxic as well, whenever it was alone or whenever it was combined with other chemicals and heavy metals. And by calling this a genotoxic, it was because that it induced the DNA breakage or the DSB. So we need more studies on the probability of this kind of interaction, how it can cause an impact on human health and other animal or insects. And thanks to my advisor, my co-advisor, collaborator, and the beekeepers who voluntarily provide me or give me the chance to take the samples. And I'm happy to take any question. Yes.
I noticed that the number of honey samples and the number of pollen samples
Mm-hmm.
were not the same.
Yep.
Was there a reason why you had fewer honey samples than pollen?
So for pollen sample, we collected through the season, and starting from June, July, and August. and that's why we have too many pollen samples to analyze, but honey samples were collected once by the end of the season, the fresh one, which usually generated in October or December. October or September.
Did any of the hives that you sampled suffer from the colony collapse during the study?
No, they were all healthy. Yeah.
And how did you analyze the samples for the heavy metals and the pesticides?
Well, I analyzed them in soil, water, and plant lab at CSU, and for pesticide, I analyzed them at a lab in Cornell University. So they did it for me, the analysis. But it was analyzed. It's called the Quisher analysis for pesticide. They use it to find out if, to find out 92 type of pesticide, if they exist in this sample or not. Yeah. Thank you. Other question? Well, thank you, and thanks for having me.